Hello everyone, hope you all are doing well. So today we are going to do topic 55 of paper 2, and here we will be discussing plant growth substances. Plant growth substances. So what are plant growth substances? These are substances which are synthesized These are substances which are synthesized in particular cells and are transferred to other cells. Synthesized in particular cells and are transferred to other cells where in extremely small quantities influence the developmental process. So plant growth substances which are synthesized in particular cells and are transferred to other cells in very extremely low quantities, in very extremely small quantities, influence the development process, and in only small quantities they influence the development process. So this was the definition of plant growth substances. Now moving on, what are the applications and commercial use of PGRs, that is plant growth regulators, or you can say substances regulators? There is slight they're kind of same thing. Application / commercial use of PGRs. So we are discussing here commercial use of PGRs. First of all, rooting and plant propagation. They are used in rooting and plant propagation. Auxin, like IBA, there are examples of auxin: IBA, NAA, 2,4-D, 2,4,5-T. So these are used here. IBA produces a strong fibrous root system. IBA produces a strong fibrous root system. Now secondly, they are very helpful in germination and dormancy. So here GA, that is giverlic acid, promotes germination. GA, that is giverlic acid, it promotes germination. ABA, that is abscisic acid. ABA, it is germination inhibitor of germination, that is it stops or reduces germination, inhibits germination. You've got auxin, it is helpful in breaking of dormancy. Some giverlin is also helpful in breaking of dormancy. Auxin and giverlin, breaking of dormancy. Now they are also helpful in fruit set and development. Fruit set and development, such as for fruit setting, 2,4,5-T is very helpful. In grapes, fruit size is increased by giverlic acid, that is GA. Shelf life increment of fruits and flowers. Life increment of fruit and flowers is done by cytokinin. Or and another feature is that parthenocarpic fruit. It is done by GA, IAA, that is IAA and PAA. So these help in parthenocarpic fruit. Now moving on, sex expression to next point, that is number 4. That is sex expression. Production of male flower. Male flower is produced by GA. You can say this in cucumber. Female flower is produced by auxin, and this can be also seen in cucumber. And some female flowers are also produced by GA in maize, with the help of GA in maize. So they help in sex expression. They also help in abscission. So NAA and IA all help in induction of abscission. They all induce abscission. Morphogenesis. These auxin and cytokinin have a role in morphogenesis. Weed control. By 2,4-D, 2,4,5-T. Plant organ size. So plant height is increased on this pH. Each plant height is increased by GA. IBA shortens plant height. It helps in some shortening of plant height. Cytokinin increases tillering by cytokinin. Moving on, anti-transparent. That is anti-transparent are ABA and PMA. That is, they reduce transpiration. Papaya latex flow. It is done by ethiphone. Rubber latex flow. It is done by 2,4-D and 2,4,5-T. Let me rough little bit. Number 12, fruit ripening. Fruit ripening, it is done by ethylene, that is ethyl. Sugar cane ripeners. These are glyphosate and CCC. These help in sugarcane research. So these were the commercial use of PGRs, that is plant growth regulators. Now some differences was asked in previous year from this topic, so we will discuss it. First of all was difference between phytohormones and PGRs. Phytohormones and PGRs. So phytohormones are natural, whereas PGRs are synthetic. There is a slight difference. So they are mainly similar, but there is slight difference you should know it. Are maintained in low concentration by plant. Maintained in low concentration by plant. These are not maintained by plant at low concentration. At low concentration, so not because they are maintained in low concentration, so that they don't show toxicity or don't act as herbicide or don't act as herbicide. Their overdose acts as herbicide. Overdose acts as herbicide. Here you can give example such as IAA. If IAA will be given in high amount, it will give act as herbicide. And there are 5 categories of phytohormones that are you're almost knowing this: auxin, gibberellin, cytokinin, ethylene, and abscisic acid. Not abscisic acid. And here the example is IBA, or you can say cyclolel. And a similar type of question was asked in one more time, that is difference between plant hormones and synthetic plant growth regulator. Difference between plant hormones and synthetic PGR, that is plant growth regulators. So plant hormones are chemicals which are synthesized by plants naturally. These are synthesized by plants naturally. These are synthesized naturally by plants during their metabolic purpose. And synthetic PGRs are synthesized artificially by human to regulate plant growth and development. Artificially to regulate plant growth and development, as they are natural. They are natural, as they are naturally applied, so they can be natural, and these are applied to plants by humans. These are applied by humans in very low amount, present by regulation by plants. In very low amount, or you can say low concentration, present in regulation. Used in high amount to increase production. It is used in high amount to increase production, growth, yield and development. And you must know that these synthetic PGRs mimic the function of natural plant hormones. These mimic the function of natural plant hormones. So these were the differences that you must know, as in previous years. Very similar, both of them. Now moving on to the next point, or you can say not next point, next topic of this session, that is functions of phytohormones. Functions of phytohormones, that is plant growth hormones. So first one is auxin. It is used in cell elongation and stem elongation. Cell and stem elongation, influences root formation, responsible for apical dominance and photoperiod. Responsible for apical dominance and phototropism. promotes xylem differentiation, promotes xylem differentiation, retard leaf abscission. It promotes retards leaf abscission, promotes fruit growth. So these are some of the common functions of auxin. Now moving on to number 2, that is gibberellin. We will discuss gibberellin in detail because it has been asked many times. Gibberellin is one of the favorite questions from this topic by UPSC in agriculture optional. So I will discuss it in detail. Gibberellins. So gibberellins were isolated from Buchan or fully settling disease of rice. It was isolated from fully settling disease of rice. There are 6 gibberellins for there are mainly 6 gibberellins isolated for fungus 6 gibberellins from fungus gibberella. These are GA1, GA2, GA3, GA4, GA7 and GA9. 1, 2, 3, 4, 7 and 9. And 3 gibberellins were isolated from bean seeds and 3 from bean seeds. These are GA5, 6 and 8. These are GA5, GA6 and GA8. Most commonly available is GA3. GA3 is most commonly available. Now, what are the some of major functions of gibberellins? What are the functions of gibberellins? So they are first of all helpful in stem elongation. They increase length of internode. Suppose there are 2 nodes, this is internode, so increase length of internode. It also speeds up RNA synthesis, which helps in stem elongation. It converts dwarf plant to normal height. Converts dwarf plant to normal height. Example, rosette plant of sugar beet. This is rosette plant of sugar beet. Third, substitutes cold treatment. Many biennials complete their life cycle within single year by treatment of gibberellin. Biennials complete their life cycle in 1 year, in single year, after they have been treated with gibberellin. So it substitutes cold treatment. Parthenocarpic fruits. Gibberellin also induces parthenocarpy. In tomato, it can be seen introducing parthenocarpy in tomato, apple, pear, etc. And it induces more efficiently than auxin. Number 5, it breaks dormancy. Example, in potato. Promotes flowering in long day plants and induces maleness. Promotes flowering in long day plants. Also it induces maleness. Maleness. Moving on. Number 7. Increases the size of leaves of fruit. Leaves and fruits increases size of leaves and fruits. It prevents senescence. It increases cell division and cell size. Increases cell division and cell size. So these are some of the functions of gibberellin. Now moving on to third one, that is cytokinins. And now some important functions of cytokinins are cell division, tissue culture, used in situ tissue culture, and it delays leaf senescence. That is, this one is senescence. Now for function of ethylene. It helps in fruit ripening, leaf and flower senescence, this one senescence, asmic senescence, leaf and fruit abscission, It also in floral sex determination, in monoecious species. So these were some of the advantages of ethylene. Now moving on, what are the some advantages of abscisic acid? Let me have this. Number 5, advantage or you can say the functions of abscisic acid. Stress response, they help in stress response, stimulate stomatal closure, that is why they reduce transpiration. Stomatal closure, inhibit premature germination of seed, premature germination of seed. It also helps in embryogenesis. Seed dormancy maintenance. So these were some of the functions of abscisic acid, and these were the functions of all 5 phytohormones or plant growth hormones. Now moving on to the next topic of this session, that is physiological functions of phytochromes. Physiological functions of phytochromes. So phytochromes are class of photoreceptors. These are phytochromes are class of photoreceptor in plant. This is a class of photoreceptor in plant, bacteria, fungi, used to detect light. Detect light. Their role is, or you can say their function is, germination of seed, which is also known as photoblasty, synthesis of chlorophyll, CHL means chlorophyll, elongation of seedling, shape and size and number and movement of leaves, shape, size, number and movement of leaves, that you can say leaves move towards light, timing of flowering in adult plants, timing of flowering. So these are the major roles of phytochromes. So I hope you all understand this, because they are all germination of seeds, synthesis of chlorophyll, elongation of seedling, movement of leaves, shape, size of leaves, timing of flowering. These are all light dependent factors. So there's where the function is of phytochromes. So that is all for today. For any queries, do comment. If you like it, please press the like button, do share and subscribe, keep making notes. Thank you, have a nice day.